So Björn, I hope you're with us, and very nice to see you, at least on screen.
Good to see you. Thank you, Uri, for the kind introduction, and thank you, Ilya, for putting this conference together again. I'm really sorry I can only join you by via Zoom, only virtually, and I most certainly hope that I will soon be in person with all you guys again. I'm going to talk about the master regular genome stability and how aging clocks function. I want to start with a remark on the evolutionary history of aging, where aging is, in our opinion, the default state of any life on Earth. The problem that even the simplest replicators really faced was spontaneously they would be disintegrating, they would be damaged, they would be subject to entropy. So aging really is the default, and what had to be evolved is actually the ability to maintain the genetic information. And this task has been really divided and taken over by the germ cells, where the germ cells indefinitely maintain the genetic information. And because they essentially can do that and up until infinity, the soma only needs to be maintained for as long as it takes to generate the next generation, upon which the soma can really age, decay, and die. And that's bottom line, basically why we age. And our maintenance only needs to last for a single lifespan, for a single generation. So we wondered whether if there are naturally very high levels of DNA repair capacity in our germlines, could we actually transfer them to maintain the genome in the soma better? Now, of course, the soma and germline very complicated in humans. We turn to our favorite model, C. elegans, as a model to understand the dichotomy of the germline and the soma. And we knew already that some of the DNA repair systems are exclusively present in the germ cells, like homologous recombination repair, global genome nucleotide excision repair. And we wondered, can we confer that to the soma? A highly talented PhD student, now postdoc in the laboratory Buchabal asked the question whether there can be a general mechanism that could curb the DNA repair capacity in the soma as opposed to the germline by looking at the promoters of DNA damage response genes in C. elegans, and he identified on the majority of such genes the CDECHR motif. And the CDECHR motif is known to be bound by the so-called DREAM complex, the dimerization partner RE-like proteins E2S-MFB core. That is a transcriptional repressor, and we knew for a long time that this is formed in C. elegans in the soma, but not the germline. And we then hypothesized that this DREAM complex could curb the expression of DNA repair genes. Our prediction would be that when we now mutate components of the DREAM complex, DNA repair gene expression should be de-repressed in the soma, and they should confirm resistance to DNA damage. And this is exactly what we observed. Here we take the L1 stage of animals. These are 90% of all cell divisions have already been completed. They just grow in size then until adulthood over the next 2 days. When we, however, treat them with DNA damage, in this case UVB, they are delayed in their development. We can see that here at 2 days after they were treated, all the worms in the UV treated are still in their larval stages, while the untreated have completed development, they are adults, and all of the DREAM mutants, Lin52, DPL1, EFL1, Lin35, are resistant to DNA damage. They can grow better. There's a bona fide function of the DREAM. When we look at double mutants of 2 components, they're epistatic, they have the same degree of resistance, they grow better after DNA damage. Do they repair better after UV? Thymidine dimers are formed that we can visualize with the cyclobutyl pyrimidine dimer antibody, and we see them formed in the slot plot here after UV. And 24 hours later, where there's inefficient repair in wild type, they are very effectively repaired in Lin52 DPL1 mutants. dream mutants repair better. As we found these dream regulatory sites in the majority of DNA repair genes, we asked whether by RNA-seq comparing Lin52 dream mutants versus wild type, we can actually see DNA repair genes being de-repressed. This is what we see, consistent with mutating a repressor. Many genes are induced, and among those, we find many DNA repair genes that are actually operating in a nucleotide excision repair, interesting cross-link repair, base excision repair, homologous recombination repair, mismatch repair, non-homologous, and joining all these different repair mechanisms, leading us to hypothesize that maybe these animals become resistant not to UV, as we just showed in the previous slide, but to all types of DNA damage. This is what we observed. Ionizing radiation, we need here very high doses because it's a post-mitotic cell types. Dream mutants are resistant. This depends on non-homologous end joining. CQ70, essential factor of non-homologous end joining, precludes the induction of resistance by dream mutants. Alkylating lesions, MMS-induced, we see a significant resistance by dream mutants. Even a highly sensitive Pol-eta translesion synthesis Pol-eta mutant that is hypersensitive to MMS can be conferred resistance by dream mutants. Cisplatin interesting crosslinks repair very, very different lesion type, different repair mechanisms. We see resistance. Now you might argue, okay, this is all in worms. What do I care? I'm a human. Does that help me in any way? Now the dream complex is highly conserved. It's exactly the same protein complex in worms as in humans. And in humans, dream targets are known by ChIP-seq analysis. And we find indeed that there's a significant enrichment among dream targets of human DNA repair genes. Does this now also work in humans? Now in humans, everything's more complicated. Different components of the dream can operate in other complexes, making their knockout rather complex. But we know that in humans, the DYRK1A kinase regulates the assembly of dream by phosphorylated Lin52. When we inhibit DYRK1A, either by the natural compound harmine or the chemical inhibitor INDI, then dream disassembles, and we hypothesize that this could then also lead to the de-repression of DNA repair genes and could provide a rationale for lowering the accumulation of somatic mutations, thus lowering cancer risk and promoting longevity by maintaining the genome stability of the genomes longer. Does this work? Here we now treat human quiescent cells. These are U2R cells in quiescence because their dream is very active. And we inhibit the DYRK1A either with harmine or with INDI, and we see de-repression of dream target genes. So it indeed inhibits dream. And among those, we find many DNA repair genes. Does this now lead to DNA damage resistance? And indeed, we find this. We treat the cells with UV or with MMS, alkylating lesions, UV-induced lesions, completely different damage type. We see normally a high apoptotic induction of apoptosis that can be prevented by inhibition of dream with a harmine or in both cases. Can we take this to an in vivo situation as a proof of concept? We use the ERCC1 knockout mouse as a model for macular degeneration, where this is a repair defect that in human and mice leads to premature aging. And we see that in these retinas, we see already after 2 weeks apoptotic cells, the retinal degeneration in the outer nuclear layer, and we can prevent that by the treatment of harmine, significantly prevented. And we see indeed that the accumulation of DNA damage visualized with gamma H2AX that occurs in the retina in the inner nuclear layer that spontaneously now accumulates in the repair deficient mice can be prevented by harmine treatment. So I showed you here that dream mutants in C. elegans elevate resistance to DNA damage by improving repair kinetics, that these dream mutants de-repress somatic expression of DNA repair genes and confer germline-like repair capacities because these repair mechanisms are usually present in the germline. The dream complex inhibition in quiescent human cells elevates resistance to distinct damage types. We can take this in vivo in a mouse model for retinal degeneration where dream inhibition protects. And we proposed that dream inhibition offers a pharmacological route for enhanced genome maintenance to prevent DNA damage-driven cancer development and aging. Now I want to switch gears for the last few minutes and tell you a bit more what are the prerequisites to take this now into anti-aging medicine. So we need preventive treatment that target causes of aging-associated disease and the functional decline, and we propose that the dream regulation of repair capacities is such a really causal intervention. But to really implement that clinically, we also need biomarkers that assess biological aging and predict the efficacy of such anti-aging treatments. Now, if we want to measure aging, it is important to realize that every human ages slightly different. Some age faster, some age slower, as Uri also just had pointed out in his presentation. And we need to quantify biological age and not only chronological age. Steve Horvath has really pioneered the field of biomarkers of aging by generating methylation clocks that are based on the changes in CpG methylation pattern that you can readily quantify. And among all these plethora of methylation sites we have in the human genome, there are some that correlate very well with chronological age that can then predict the age of an individual. Now, the question that arose is really that this accuracy of aging clocks that looks to many like it must be a program because it's so precise, is that compatible with the evolutionary theory of aging? Indeed, methylation data sets and also other types of data highly correlate with chronological aging that allow the biological age to be derived from deviations from this chronological age. And does this now mean that there would need to be a program? While evolutionary theory predicts that aging is caused by the vanishing selective pressure on somatic maintenance, because as I pointed out, only germ cells need to live forever, not the soma. And so essentially what we are putting forward is that even the stochastic process, the pure accumulation of stochasticity could provide an aging clock. And the way we did this, David Meyer in the lab, an outstanding bioinformatician, the way he conceptually addressed this issue, whether stochastic variation alone can provide aging clocks, is that he defined a ground state. A ground state means the age 0 at the beginning of life, and that every biological organism has at the beginning of its life. And then the simple thing we did was just to add stochastic variation on the ground state. The ground state are just numbers, random numbers computationally generated. And for H1 would be one round of stochastic variation. H100 would be a 100 times stochastic variation added to the ground state. And we defined 3 training data sets of this that we then took for an elastic net regression to then ask, can we from this training data set predict 3 data sets that we independently generated from the ground state with purely stochastic variation? When we do this, we see an age predictor that does not predict anything. It doesn't change, it stays stable. Why? Because if it's just variation, the negative and positive cancel each other out, but this biological can either have a unmethylated or methylated CpG. You can have a gene that is not expressed or expressed, but none of that can be negative. So we need to define limits of this variation that cannot go below 0. It doesn't make sense. And so we define the variation between 0 and 1, because every biological parameter can be normalized between a value between 0 and 1. And then we get a nearly perfect age predictor based on stimulation of purely simulated data, a ground state with stochastic variation. Then we wondered whether we could take this to methylation clocks. So we virtually defined methylation, a pattern, by defining a ground state of assumed 1,000 single cells. And in each of these 1,000 single cells, for each site of methylation, virtual methylation site, we define a value of 0 unmethylated or 1 methylated. And then, for example, a ground state of 0.13 for a given methylation site would mean that in 130 of those 1,000 cells, this site would be methylated, and in 870, it would be unmethylated. Then we ask them to randomly flip each state in each cell based on the maintenance efficiency and the de novo methylation that each site can be characterized by. And these we can fix, these we can assume biologically, but this will be the degree of the random variation, the stochasticity we add. And then we just average them and define from that an aging clock. And we can generate a perfectly prediction from simulated age to predicted age here with a fixed maintenance rate. What determines the pace of aging is the maintenance efficiencies. Here we define different maintenance efficiency that define how much stochastic variation there is. If there's nearly perfect maintenance, then there's no change, so age would not change. When there's very little maintenance efficiency, the age predictor would increase very, very rapidly. So the degree of maintenance is the determining factor. We can use this by in real data, real biological data. So here we ask, can we predict the age of nearly 400 human blood samples by just using one blood sample from the youngest one as our ground state, and then do nothing else than for each age of each year of age, add a round of stochastic variation, and we can predict the age here. We can take that to over 11,000 independent samples from 50 different data sets and significantly predict age. We can use this actually to make aging clocks in all mammals, where the only biological data we use is from the youngest sample. It's a dolphin. We set that as ground state, then only add stochastic variation. And here are the R values that allow us to predict age in all these different mammalian species without any additional biological input. Can we measure accelerated and decelerated aging? Because that's the purpose of it all. And indeed, here 3 models of delayed aging in mice, 2 genetic model and calorie restriction. We see a significant deceleration of aging with our stochastic clocks for different ones, just different maintenance efficiencies, we assume. And we find accelerated aging in humans that smoke. We see decrease in biological deceleration of biological age and heterochronic parabiosis, where an old mouse is connected to the circulation of a young mouse that prevents aging. And in a model of fibroblasts that were by the Yamanaka factors reprogrammed, we see over the course of 28 days, we see a reversal of its biological age. It's rejuvenating based on the stochastic clocks. So what I showed you here, the last part, is that aging clocks can be based on purely accumulating stochastic variation. Simulated stochastic variation provides an accurate chronological and biological age prediction. Any data type with a ground state of age 0 can provide an aging clock. It doesn't matter whether it's methylation or any other parameters. Age-dependent changes in biological parameters are driven by increased stochastic variation. The degree of this stochastic variation determines the pace of aging. Geroprotectors that could reduce the variability and restore the regulatory tightness could be very effective. And the accuracy of the aging clocks is fully compatible with the evolutionary theory of aging. I thank you very much for your attention. I thank the people who did the work, and the mouse work was done by George and Kalina in George Garina's lab. I acknowledge all the funding sources. If you're interested in this type of work, we're recruiting postdocs, As the very last thing, I just want to encourage you to register and join us for the Aging Genome Conference in Heidelberg in 2025. Thank you very much for your attention.
Thank you very much, Björn. Any questions? So, Tomer, yeah?
I want to ask about the dream complex and if it has any trade-offs like reduced fertility.
Okay, so the question is, does inhibiting the dream complex have any trade-offs such as reduced fertility? Björn?
Excellent question. So we find that in C. elegans, these Lin52 mutants have a very slight reduction of fecundity indeed. So this would be consistent with now resources from the germline would be expanded into in the somatic maintenance, and that could be a negative effect. That is very well possible. We have now generated dream mutant mice where we will systematically investigate to which degree here a reallocation of resources to somatic maintenance is happening. Very good question.
Okay. More questions? Yeah, there's a question from back there. I'll repeat, yeah.
So you demonstrated very nicely that you can create epigenetic clocks from just stochastic noise, but some works say that you can also create them from non-stochastic events. When do you think each one should be used and what are the advantages?
So the question is, you have shown elegantly that epigenetic clocks can be based on stochastic effects, but there's also the possibility that they can be based on non-stochastic effects. When should each form be used? Did I understand your question? Yeah.
Yeah, so of course you could always derive a program for anything. I mean, anything that changes linearly with age, you could propose a program. But the point here is really that stochasticity itself is sufficient. That is really the important message to give this age predictor. And of course, there's an underlying mechanism why stochasticity increases. By default, it would increase. But by default, it would increase very, very rapidly. Without our maintenance, we know from human DNA repair deficient patients that they age within a decade of life. That is the stochasticity, which is driven, for example, by DNA damage, is just unrelentlessly accumulating. So the critical program are the DNA repair mechanisms and the maintenance mechanisms, all these cellular maintenance mechanisms that we require to curb the normal entropy that just would take us apart immediately if it wasn't for our maintenance programs. So these programs are really the decisive ones, and these programs we need to target for really effective interventions to keep the force of stochasticity in all our biomolecules at bay.
All right, Valerie, could you come up? So we're happy to close. Björn, thank you very much. We can hope to see you soon in person.